This method can help answer key questions regarding the mechanisms and energetics of protein transport across chloroplast membranes, as well as the location of proteins within plastids. The main advantage of this technique for thylakoid protein targeting is that it allows for complete control of the thylakoid's environment during transport, including temperature, pH, ionic conditions, and precursor concentrations.
Begin by soaking approximately 55 grams of peas in 400 milliliters of distilled water for 3 hours. Sow the soaked peas in a plastic tray in soil covered with a thin layer of vermiculite and grow for 9 to 15 days as described in the text protocol. On the day of chloroplast isolation, prepare a continuous density gradient by centrifuging a mixture of 15 milliliters of percol and 15 milliliters of 2xGB buffer in a 50 milliliter polycarbonate high-speed centrifuge tube at 30,900 Gs in a fixed-angle rotor for 30 minutes at 4 degrees Celsius with the brake off. Then harvest approximately 25 grams of 9 to 15-day-old pea shoots. Add them to 95 milliliters of cold 1xGB buffer in a chilled blender with sharpened blades and homogenize. Filter this mixture through a minimum of 2 layers of cheesecloth placed in a funnel over a chilled beaker. Centrifuge the filtrate using a 50 milliliter polycarbonate centrifuge tube in a swinging bucket rotor at 3000 Gs for 5 minutes at 4 degrees Celsius. Discard the supernatant and use a paintbrush to gently resuspend the pellet in 1 milliliter of 1x.